don't want to do this anymore. Okay, so you guys, just for the movie, this is Wednesday, November 20th, and we're going to be going over our Chapter 17 metabolic test results. Business, you guys, we do have open lab this Friday from 8 a.m. till 10 a.m. And very importantly, you guys, Monday, what's going on Monday? The lab exam, good. And folks, we'll start a little bit later than normal. We'll start around 8:30 a.m. So please try to be here at 8:30 a.m. And then if you're starting earlier, just come early, all right? And then folks, also do remember we're turning in our lab exam study guide packets with lab results. So last lab, we passed out a checklist to make sure that you would include everything. If in doubt, you guys, just include it. Just staple it to your packet. And then I'll use that checklist just to give you points. And folks, I will get those packets back to you right after Thanksgiving break, because folks were reminding me a lot of their notes are in those packets, and they're like, how are we going to do the unknown without our notes? So I'll try to get them back to you right when we get back from Thanksgiving. We're going to start the unknown right after Thanksgiving, and I've got a packet of information for you folks that will tell you what you're going to do each day of the unknowns, okay? But make sure you have your PPE. You'll need that every day. Okay. So I think that's all, excuse me, I think that's all of our business. So folks, what I'll do is break this up into little chunks with regard to the movies. We're first going to go over the IMViC tests. I'm going to tell you how to read them. One of them you have to add reagents to. So I'll go over kind of like the background, and then we'll break and let you guys read your tests. And then as a class, we're going to put the class results up here. So first of all, folks, with the IMViC, let me just quiz you. IMViC is actually 4 different tests. So what does the I stand for? Indol, right. So you guys, if we have a positive indol test, what does that tell you about your microbe? This would be the interpretation. Exactly. If your microbe makes indol from tryptophan, it means your microbe makes the enzyme tryptophanase, right? And you guys, how did you run the indol test? How did you do that on Monday? Do you remember how you ran it? The dry slide, right? Exactly, you guys. Remember, it's nice, quick, fast, benchtop test. So folks, if your microbe made indol, what color did the dry slide reagents turn? Red, good. So remember that. Red's a positive indol test. And if the indol dry slide did not turn red, what does that tell you? It's a negative indol test. And what does that tell you about your microbe? It doesn't make tryptophanase. Exactly. So you guys, that's the interpretation. So the appearance is how does it look? Like what color it is. And then I want you to tell me, is that positive or negative? And the interpretation, the significance. What does it tell you about your microbe? Good. Folks, what does the M in IMViC stand for? Methyl red, good. And folks, if we have a positive methyl red test, what does it tell us about our microbe? Yeah, mixed acid fermentation, a very specific fermentation pathway. So you guys, you remember you inoculated MR broth, which is high glucose broth, right? So when you pull it out of the incubator, are you just going to look at it and determine if it's positive or negative? No, what do you have to do? Yeah, and you guys, this is such a common mistake. People forget to add the methyl red pH indicator, which we have up front. So after incubation, you're going to add 5 drops of the methyl red pH indicator. The reason we can't add it ahead of time is why? It'll kill the microbes, right? So we have to let them grow and ferment and then add the methyl red pH indicator. Now, folks, it's easy, for me, it's easy to confuse phenol red and methyl red. Methyl red pH indicator turns red at really low pH, pH below 4.4 to 4.8. So you guys, if your microbe carried out mixed acid fermentation, it makes all these acids, it's a really, really low pH. So when you add your methyl red pH indicator, what color would a positive test appear? Red, exactly. Good, you guys. And then if your microbe didn't carry out mixed acid fermentation, when you add the MN indicator, it won't be red, okay? So far, red is positive for both indol and for methyl red. Good. Folks, what's the V of IMViC stand for? Vokes-Proskauer, and this is a test for, it's a big name, you guys, butylene glycol fermentation, also known as 2,3-butanediol fermentation. Now, I'm going to run that as a demo because the reagents are really toxic. Okay, but it's very similar, the procedure is similar to the MR test. We inoculate VP broth, and again, it's just high glucose broth. And then after incubation, I'm going to add 2 toxic reagents, and that's why we're doing it as a demo. I'm going to add alpha naphthol and 40% potassium hydroxide, and we do this in the safety hood. So folks, if your microbe carried out that butylene glycol fermentation, there will be a unique intermediate. The long name is acetylmethylcarbinol. The short name is acetoine, right? So if your microbe carried out butylene glycol fermentation, it should make acetoine. When we add our alpha naphthol and 40% potassium hydroxide, within 30 minutes a red color will develop. So again, you guys, red is a positive VP test. If your microbe has a positive VP test, what does it tell you about them? They carry out butylene glycol fermentation. Good. Another fermentation pathway. Good job, you guys. Folks, what does the C in IMViC stand for? Citrate, right. So the citrate test is a test to see if your microbe can use citrate as a sole source of carbon and energy. Now there's different citrate media. The one we use, I love it, Simmons citrate agar. Which pH indicator is present in Simmons citrate agar? Great exam, lab exam question. Bromethymol blue, good. So folks, if your microbe can use citrate as a sole source of carbon and energy, the final pH will be what? Acidic or alkaline? Alkaline, good. And what color does bromethymol blue turn at alkaline pH? Deep blue, beautiful blue color. Okay, so folks, what we'll do is we're going to stop the movie here, and then I'm going to give you like 5 minutes, kind of get organized, to read your results, record your results, and then I'm going to ask you, as a class, what our results were for E. coli IMViC and Enterobacter aerogenes IMViC, and we're going to discuss why are these tests helpful, right? And what I want you to remember for lab exam too, these tests are really helpful in letting us distinguish between E. coli, which you guys might remember, we said high concentrations of E. coli in drinking water or food is an indicator of what? Fecal contamination, right? So we want to find out if we have lots of E. coli in our food or drinking water, as that indicates possible fecal contamination. So the IMViC test, you guys, what it will do, it will let us distinguish E. coli from its close cousin, Enterobacter aerogenes, right? And Enterobacter aerogenes, although it can grow in the intestinal tract, if all we're finding is high concentrations of Enterobacter in our food or drinking water, it doesn't necessarily indicate fecal contamination, right? Okay, so we'll stop the IMViC intro here, and then we'll let you guys get your full PPE on, go grab your IMViC test, set them up in the your test tube racks, score their appearance, record their appearance, and whether the appearance tells you they're positive or negative, and then we'll put our class results up on the board. Okay, all right.